Let's look at level 2. Recall that level 2 involves internal defenses. In this case, the microbe has breached the surface barrier. This is going to involve antimicrobial proteins, and we'll look at these in greater detail later. I'm just going to list them now, but it involves complement,
cytokines, such as interferons and interleukins, phagocytosis,
inflammation, and fever. Let's begin with complement. Complement consists of a set of plasma proteins. Once the first complement is activated, it initiates an enzyme cascade where you have the first enzyme, remember enzymes are proteins, that is activated. And when it is activated, it is cleaved, which means it breaks apart into two molecules. One of the molecules will function as an enzyme to activate a second complement protein or plasma protein. The same thing will happen. That protein will be cleaved, and one of the molecules then will function as an enzyme for the next reaction. So it's a cascade event where the activation of one of the plasma proteins will be used to activate the second one, which will then in turn be used to activate the third one. So just understand that it is an enzyme cascade consisting of plasma proteins. There are three outcomes. You will need to know the three outcomes, and there will be three pathways, which we'll talk about in just a minute. The first outcome is what we call opsonization. This is a new term for you, but complements can function as opsonins. Opsonins are molecules that will bind to cells that need to be phagocytized, and they enhance or make it easier for the phagocytic cell to eat the cell that has the opsonins on it, or let's say if it's a bacterium. So the complement will bind to the bacterium, and that will make it easier for a phagocytic cell such as a macrophage to eat the bacterium. This illustration is of opsonization. The green cell is the phagocytic cell, the red rod is the bacterium, and the purple ovals are the complements. So the complements will bind to the bacterium, and that will provide a binding site for the phagocytic cell. In order for phagocytosis to occur, the phagocytic cell must recognize and bind to molecules on the surface of the cell that is to be phagocytized. Complements will function as those molecules. So now you can see the phagocytic cell binding to the bacterium with the complements. So the phagocytic cell will bind to the complements which are attached to the bacterium, and this will initiate phagocytosis. And the bacterium then is brought into the phagocytic cell. The bacterium then will be digested. Let's continue with complement. The second outcome is the fact that complements can enhance inflammation. They do this in several ways. They can function as positive chemotaxic chemicals. Let's break this down. Positive refers to the fact that leukocytes will be moving toward the chemicals. Taxic refers to the movement, and chemo refers to the chemical. So positive chemotaxic chemicals help draw leukocytes, such as neutrophils, to the area. In addition, complement proteins stimulate basophils and mast cells to release histamines. Histamines increase vessel permeability. The last outcome is the formation of membrane attack complexes. Complements come together to form pores in the membranes of the pathogen, such as the bacterium. Once the pores have been formed, these membrane attack complexes are formed. Water diffuses into the cell by osmosis. When the water moves into the cell, such as the bacterium, it will cause the cell to lyse. This figure illustrates the three outcomes for complement: opsonization on the left, enhancement of inflammation on the right, and down at the bottom, the formation of the MAC or the membrane attack complexes. There are 3 pathways in complement: the classical pathway, the lectin pathway, and the alternative pathway. Let's take a look at the 3 pathways and what triggers each pathway. So you'll need to tell me what activates the first complement in each one of the 3 pathways, similar to what you had to learn for coagulation. We're going to start with the classical pathway. It was discovered first. And the term complement was used because when it was first discovered, when the complement plasma proteins were first discovered, the proteins were seen to complement the antibacterial activity of antibodies, hence the term complement. So what triggers the classical pathway? It is when a complement protein, the first one in the cascade, comes in contact with an antibody or antibodies that are bound to pathogens. For the alternative pathway, it's called alternative because it was discovered later. What triggers the first complement here are just simply the pathogens or their products. So it could be a toxin that they produce. And the third pathway is called the lectin pathway. Lectin, a plasma protein, is going to bind to polysaccharides found only on microbes. And when this occurs, this is going to trigger the first protein in the cascade. Recall, all 3 pathways lead to the same 3 outcomes.